Good morning, everybody. Thank you so much for taking time and joining us this morning. Before we start, I would like to, on behalf of Shimadzu, we would like to thank you all for taking time joining this morning. A big thanks to MBC Biolabs for giving us an opportunity to place our system here at MBC. And we hope you get to use the system to the best of its abilities. Your feedback will be extremely valuable, and we hope you get fantastic data out of it. So this seminar, it is about our low pressure bioinert prep LC system that we are placing here at MBC Biolabs. Here's a brief overview of the presentation. We want to talk a little bit about Shimadzu, about our analytical systems, what are the tools that we have for our pharma and biopharma customers, and finally, we want to spend a lot of time talking about our FPLC, our bioinert semi-prep LC system that we have placed here at MBC. A brief history about Shimadzu, if you don't know about it. It was back in 1875, Genzo Shimadzu Sr., he started the company to manufacture educational equipment for physics and chemistry experiments. A few years later, he started manufacturing a hydrogen balloon and he successfully launched manned flights. In 1895, the company started manufacturing storage batteries, and in 1996, they succeeded in producing radiographs. Shimadzu Corporation is a global company. It is headquartered in Kyoto, Japan. We have a huge facility in Japan. As I said, it's a global company. It is all over the world. We have the application development, research, R&D, manufacturing, sales, and innovation center across the globe. As of March 31st, we have about 13,000 employees globally. Shimadzu works on different business units. As you can see, we manufacture analytical and measuring instruments. We manufacture medical systems, aircraft equipment, industrial machinery and equipment. Our core business is analytical and measuring instrument that gives us the maximum revenue. The US subsidiary of Shimadzu Corporation is called Shimadzu Scientific Instruments, which we all represent. It was established in 1975, headquartered in Columbia, Maryland. As of now, we have 500 plus employees. Apart from the headquarters, we have 9 regional offices throughout the country. Each regional office has its own training, service, sales, and technical support to take care of that region. And for California, the regional office is located in Pleasanton. We are very proud to say that our manufacturing facility is located here in US. It was established in 1997, headquartered in Canby, Oregon. Here, the employees, we have 100 plus employees that manufacture our LC, LCMS, GC, GCMS, UV here in US. Our systems are class 1 medical devices. Now I'd like to hand over to Steve, who will be talking more about our portfolio of the products and the applications for pharma, biopharma.
Thanks, Dipshika, and again, thanks, Flavia, and everybody else from NBC. So one of the things that, as Dipshika had mentioned, we have a wide range of business units, but the analytical instrumentations we're going to focus on today is what we represent. And in addition to the FPLC-like system, the Bioinert system, we have a wide range of instrumentation going from chromatographic, mass spectrometry, balances, physical testing machines, and spectroscopy. So there's quite a range here, and in each one of these, there's lots of models and variations designed for different purposes. Today we'll focus more on the HPLC side of things, or at least on the chromatographic side of things. But what you can see here is that Shimadzu has been involved in the liquid chromatographic instrumentation for quite some time, starting way back in 1978, and progressed over the years to have higher and more better performance and more reliable design for the modern user of liquid chromatography. And part of that has been increasing pressure or maybe a bioinert system, ease of use. And so there's lots of reasons why these things would change, in addition to applications and also computer power that would basically allow for different purposes for chromatographic needs. And then focusing on today's current product line, we have modular systems, integrated systems, bioinert systems, and supercritical fluid systems. And all of these have different purposes depending upon where you're at, and they might be more biologically based, they might be more small molecule chemistry based. And in some cases, you might need something that's very simple in a quality control environment, and something more in a discovery environment, such as our modular system, that you might need to make flexible so that depending upon your discovery needs or your development needs change, you might need to modify the system by adding or subtracting components. And one important part, again, just pointing out those four types of systems, is that within the systems, there are different, I guess, flavors or different types. And one of those factors that comes in nowadays is pressure limitation. And as since about the early 2010s, maybe just prior to that, columns, HPLC columns started becoming smaller and smaller particle sizes, which then increases the potential for back pressure. So each of the systems now has an opportunity to have a higher limit of back pressure, going up to as much as 19,000 psi. So depending upon the chromatography and column type that you need, we can cover that through the range of HPLCs listed here. And part of the advantage of higher pressures is maybe to get the same resolution, so the same ability to separate peaks, but in a shorter amount of time. So which means that if you're time constrained, and sometimes in the discovery space you might go with a higher pressure. But if you need, something that has more visual separation, or if you have a legacy column or application, you might have to go with the lower pressure. But in any case, we can cover you from the top to the bottom. So in addition to the actual hardware, some of the key features that oftentimes arise is how the end user interacts, what they need. Hardware is great, and that's where we want our engineering efforts to basically shine, but we also have to think about how the end user interacts with that. So in many cases, the software sometimes is needed to make it easier to use. And in this case, we have software that's designed for walk-up reaction monitoring or walk-up submissions, where maybe a large lab can walk up and submit, a series of 5 or 6 samples, decide what column it's going to use, what method it's going to use, submit it, and then walk away. And then that information will be acquired while they're away, while another user can come up and put another batch of samples right behind it. So that makes it easier, so which means that it becomes a tool in a toolbox for the average medicinal chemist or scientist to walk up to. And that is on the left-hand side. Now, on the right-hand side, we have an example of our method scouting system. In many cases, later stage, you oftentimes have to come up with a chromatographic method that is designed to move into a validated environment to monitor maybe impurities or some sort of reaction stability products. And so you might need 20 or 30 components to be able to be separated, to be easily monitored. And so what this allows you to do is to go through and screen multiple columns and multiple solvents in multiple conditions in order to find the best suited chromatographic separation. And that is, again, automated system where you just find a few key parameters and then walk away, and then your data is generated as you when you walk away. But today we're also going to focus more on preparative. So this is another focus that we have systems. While our system at MBC is designed for smaller scale, we do have the capability of doing larger columns and then also improving to larger scale. And so we can have expandable fraction collectors. So which means if you need a larger number of fractions, you can add multiple beds on daisy chained, and then you can have larger pumps to have a larger capacity. flows in larger columns. And one of the key things on the right we'd like to focus out is the easy fractionation capability here, so that we can have a test run and you can quickly then determine the best signal determination in the method, so that you can do a peak detection or do a time detection. So again, making the end user's experience that much easier, so that they can walk away feeling comfortable that the instrument's going to work as they intended. So in addition to HPLC, we also have quite a mass spec suite of products. Starting off on the left, going to something as simple as a single quad, which provides a mass for something as a generic or unique mass for mass detection, along with maybe UV, fluorescence, and other detection devices. And something like this might have lipid nanoparticles, which is more recent with the mRNA vaccines. They might have to monitor lipid nanoparticle ratios. But we also offer triple quads, and triple quads are largely designed for use in quantitation, but can be used for a wide range, including the single quad capability, scan functions, and a variety of other discover functions. But the big advantage of a triple quad is that it has wide dynamic range, so that you can look at very low level to high level on the same instrument in the same method. We also, then moving over to the right, we have our QTOF LCMS 9030, designed for high resolution, high mass accuracy instrumentation. In many cases, this is designed for both discovery, but also in the later stage of drug development. So sometimes you're looking for target engagement, or in this case, an example of looking at amino acid sequence determination using a peptide map for a large protein. And also on the far right, we have the MALDI 8020 OmegaTOF. And this is a system that's designed to look at large molecules, and that very simple, very fast. It's a spotting technique, mixing with a matrix, and it can be an example where maybe you've done a fractionation with a FPLC system like ours that's in the lab today. And then you want to verify what's in the fraction, what mass is in there, how pure it is. You can quickly spot that on a plate, put it inside the MALDI, and within a matter of minutes, have a mass spectrum. And this is an example of some of the applications related to that high mass MALDI OmegaTOF system, where in the case of on the left, you have an IgM, which is around a megadalton, so 1,000 kilodaltons of molecular weight, and so you can see the singly charged ion inside the spectra, along with some multiply charged species. And then you can also see the on the bottom left is the antibody antigen, which is cross-linked, so that it basically stays covalently bonded. But you can see the presence of, in this case, the antigen, the antibody, and then the antigen antibody complex. So it allows you to be able to see large masses in a very quick analysis. In addition, we have on our website, you can go to the biopharma website on the Shimadzu system, or the Shimadzu website, and there's quite a few applications related to other biologics and as well as small molecules inside there. So, and just this is an overview to provide you understanding of the instrumentation as well as the application capabilities. And while we're here over the next couple of days or via our email, you can definitely reach out and find out more about this. Now I'm going to pass it over to our tech support for the Northern California region, Andrew Meisner.
Hello, everybody. This is Andrew Meisner. Yeah, I'm tech support here for the Northern California region, and I'll be here at NBC doing training later this afternoon as well as tomorrow, and just going to be working through the entire placement process. Starting off here, we have a nice picture of the system here at the NBC Biolabs. We have a couple of different modules to talk about. In the first left stack, we have an auto sampler that has come standard with 4 ml vials. We can put lots of different types of samples in there. We have a binary gradient pump of 2 LC20AIs that are inert. There's a degasser, a reservoir tray. There's a system controller. There's complete with 2 separate detectors. We have both a UV detector and a fluorescence detector. And then we have a column oven that will provide some temperature stability for any of your samples. The large item there on the right is the fraction collector, which we'll spend a lot of time talking about today. So some key features at a glance we're going to talk about is The automation of a system like this, it allows you to load up large batches of samples, which is kind of rare in some of these other systems offered. So it allows you to run maybe, 10, 20, 30, 40 samples at a time, so you don't even have to be in the lab. Adding on to that is allowing you to have reports automatically generated at the end of each sample or at the end of each batch. We can also have automated startup and shutdown principles. So you don't even have to be in the lab, and it will start up, run a suite of startup checks, and sit there ready for you to run. In conclusion, there's also some shutdown principles so that at the end of your batch, it will safely shut down the instrument, so even after you've been long gone from the lab. Now, the other thing about a prep system like this is there are two main goals. We want to purify and we want to collect our fractions. So we need this to be as smart as possible so that we maximize purity and maximize the amount of material collected. Those should be the two main goals for everybody using a prep or semi-prep system. With that in mind, they have really increased the amount of intelligent fractionation tools for you. This means that there's a sample rescue when things go wrong, or if they go wrong. There's a 3-click parameter generation, which is about as easy as it can get. There's also and/or logic. So if you need to come up with some clever ways to collect certain analytes as opposed to not collecting certain analytes, we have a tool for that. We can also mix and match time-based as well as signal-based collection parameters. These all go into maximizing your recovery and purity, which is the essence of Prep LC. Now, this semi-prep system is bioinert completely. And with that, you can go up to over 2,000 psi with a flow rate up to 10 mils a minute. This completely bioinert flow path includes a ceramic needle, inert flow cells, and an automatic plunger seal wash to make sure that any buffers used is not going to wear on the system. Now, you have two different ways to temperature control. You can temperature control the column with your oven down to 4C all the way up to 105C if necessary, as well as the autosampler for those precious samples. If you need to keep them at a cool temperature for stability purposes, you can take it down to 4C. If you also need to have, for whatever reason, some increased temperature for maybe solubility of sample so it doesn't crash out, we can also take that autosampler up to 40C. In addition, the autosampler allows you to have lots of different types of samples. We can inject from 4 mil vials, which is what it's currently set up for. We can also have 1.5 mil vials. We can also have a special rack that can accommodate microtiter plates, deep well plates, in either 96 or 384 well format. Now on the fraction collector, this has the maximum amount of versatility. You can collect into a whole wide array of different things, including microtiter plates or vials or test tubes, and you can mix and match. It really is a wonderful type of fraction collector. Now, the first feature I want to talk about why this fractionation is so wonderful is, let's say you've spent, all week synthesizing your really important sample, and you go to inject it, and somebody maybe messed up. Maybe the sample crashed out, or the column was incompatible. Something bad happens, and your precious sample is stuck on column. Oh no, what do? Do you just turn it on and that sample's gone? No, with the FRC40, we now have a feature called the sample rescue. If something goes wrong and you need to start over again and your sample is stuck on column, we can tell the fraction head to go to a predefined area, and we can collect flow at a lower flow rate if there's blockage, and we can collect your precious sample into a designated zone, into a bottle, and then you are able to dry down and start over. This is invaluable for those really valuable samples that you may have spent a lot of time synthesizing. The next is the 3-click generation. It really is this easy. If you have a chromatogram of your typical injection volume, you load that into the simulation, you click Add, drag in how much of that peak that you would want it to collect, and click Reflect. We can see on the left-hand side here, those are the parameters that it has generated based on these 3 steps. The next time, if you were to run the exact same sample of the same concentration, it would automatically collect based on this. We also have the ability to do time-based and signal-based collection. in either or format. Sometimes, if you know a peak will come out at a very predefined moment, feel free to collect time-based. If there's going to be some variability in your samples, feel free to use a signal-based absorption, just like we had seen previously on the 3-click generation. In addition, with multiple detectors, you can choose the and/or logic to collect based on one detector or both detectors. There may be an instance where you have a compound that elicits a very strong UV response, but maybe not a very strong fluorescence response. And you can pick those. But what if you have one compound that definitely shows up on both, but you have some other compounds that you do not want to collect that only show up on one or the other? This is where the AND logic can really help you out with collecting only the compounds that you want to collect. This is a time saver. You do not want to spend all of your time drying samples and reconstituting if it's something you don't even want. The next thing is, obviously, chromatography is, well, sometimes there's some really good resolution, sometimes there's some not so optimal resolution. And we want to be able to give you the tools for every single instance. If there's some bad resolution or non-optimal resolution, what can you do? Well, Shimadzu's peak splitting tool allows you to decide, do I want to split these fractions into really pure, not so pure, and so forth for unresolved peaks? Or is it more important to you to just collect the most as much as you can? That's where you have these options in peak splitting to really give you what you want. The next is we have a slope disable. This seems a little different for some people who are maybe coming from analytical HPLC, where you want to have that beautiful Gaussian peak shape. Well, in prep and semi-prep, bad looking peaks like this are pretty much the norm. People would purposely do this, overloading the column, because the goal is to purify and collect the maximum amount of material. Peak shape is not so much a concern in this instance. When that happens, though, overloading can have some funny peak shapes that affect maybe slope trigger collection. So we have tools to override that in that instance. Let's say you have an overloaded peak where it starts and it gets the slope to trigger fractionation, but it has a small dip down just from interaction on the column eluting out of the column into the detector. You would not want to miss out on that really important big chunk of your peak. So we can disable that slope feature and collect the entire broad peak that you would want. This is just a great tool to have in your back pocket if that scenario comes up. Adding on to that, if any of these other features may seem cumbersome to you, we do have another easy add-on, which is the peak collection sensitivity. Essentially what this is doing is taking a look at if there are noisy background signals there that may elicit a slope that might trigger fractionation, but maybe you don't want to collect them, and maybe you don't have time to sit there and run a sacrificial sample to get those parameters for running the same sample over and over again. So in that sense, we can come here and use the peak collection sensitivity. And let's say, well, I know I've got 2 really important analytes that I want to collect, and the rest I don't really care about. If you know that they're going to be high concentration, let's set that sensitivity to a higher value so it ignores all of the other small peaks that you have no interest in and only collect the massive ones. That is a great quick tool to use. In addition, when you are done running a sample, we can have it linked to an automatic report format for each sample that will showcase the chromatogram. You see these little hash marks on there are the fractions that it collected, and we'll have a table below that will show you exactly where each fraction has been stored on the fraction collector. So easy access. You can go there, pull out your samples that just collected, or you can go pull the whole rack. And then you can take your report with you and find the exact fractions that you want to analyze next. These automated reports can be PDF or print out, and it's a pretty nice little feature. They are highly customizable reports as well. If you like to put an NBC logo on there, you can. If you want to add pictures or reformat any of the information, we allowed to do that. Lastly, there's some applications on these low pressure prep purifications, especially with proteins. Proteins are notorious for needing to be run at lower pressure in general. And with the size exclusion chromatography, that's always an option, as well as using fluorescence-based detection. It really is a great, what's the word? It is selective. Fluorescence is a great tool for very selective applications because while lots of things may absorb, they may not always emit again, which is very nice for when you're trying to exclude some noisy things that may also be in your sample. So we also have a wide array of reverse phase as well as other applications, but feel free to search for us. And that is all I have to say on that. And so thank you so much for taking the time to sit with us today and discuss, again, from Steve and Deepshika. And I look forward to meeting the rest of you later in the lab to do some training.
Thanks, Andrew. Thanks, Steve. Questions? Anybody got any questions for us now, or we're happy to see you in the lab?
Well, if no one has any questions, I would like to thank you, Deepishka, Andrew, and Steve, for your time and your support. And we will see you here in the community area.
Yeah, thank you so much.
Thank you.